Welcome friends. In the first part of my lecture, I talked about the structure of gill in chondrichthyes. Now, in the present series, I will be talking about the structure of gills in the teleost fishes, that is bony fishes, and I will also try to talk about the lungs. Now, to start with, we have read that the gills in case of chondrichthyes are the septate gills, that is they have interbranchial septum fully developed. On the either side of the interbranchial septum are the gill lamellae or gill filaments, which are also referred to as primary gill lamellae. The entire gill is referred to as holobranch, while as the only one lamella only on taking in one lamella only will be calling it as hemibranch, that is half gill. Now, in case of actinopterygii or teleost fishes, it has been found that this interbranchial septum is either reduced or is completely absent, which means that these two gill lamellae or the primary gill lamellae are free. They are not attached with the interbranchial septum, which in turn also means that we have increased the surface area for the exchange of gases, which was otherwise attached with the interbranchial septum. Thus, in case of bony fishes, the gills are having much more surface area, rate of respiration will be very high. Now, these gills in case of bony fishes are also referred to as aseptal gills, and again the structure remains the same, that the primary gill lamella is divided into secondary gill lamella, which are the actual site of exchange of the gases. The same again, the efferent arteriole comes with the deoxygenated blood, divides into efferent arterioles, forms capillary bed in the secondary gill lamellae, and the efferent arteriole moves out of the gills with the oxygenated blood. Gill ventilation in case of actinopterygii fishes is like this. Again, the mouth opens, but the only difference between the chondrichthyes and these are that there is no spiracle, which was one more opening for the entry of water into the branchial pouches. Here there is no spiracle, mouth opens, pharynx lowers itself, creates a sort of suction pressure, water is thrown into the pharyngeal region of the body. Second time the mouth closes and water is forced over the gills. This water is rich with oxygen and is flown over the gills, thus the exchange of gases takes place in their case. In the secondary gill lamellae, there is the counter current mechanism which optimizes the exchange of gases with the water and the gills. The accessory structures that are associated with the bony fishes for the exchange of gases are number 1 the swim bladder, and number 2 the swim bladder converted into the lungs, as is the case with the lung fishes. Now, this is the common occurrence in the osteichthyes, that there is a swim bladder which has got two purposes. Number 1, there is a air inside the swim bladder which creates a buoyancy, and this keeps the fish afloat in the water body. The other function associated with it is that of the exchange of gases. This swim bladder is actually an outpouch of the digestive system, which may either arise from the esophagus or from the stomach or from the intestine. This swim bladder has very important function here to perform, is that of it keeps some air inside which optimizes the exchange of gases. The other structure is that of the lungs. This lung is present either on the dorsal side or on the ventral side of the fish. Accordingly, we call it as dorsal lung and the ventral lung. Dorsal lung is also called as swim bladder in case of actinopterygii. The gases that are present in the swim bladder are arising either from the gastrointestinal tract, that is gut, or the gases are filled by the blood circulation. Accordingly, the names are physostomus if the gases present in the swim bladder arise from the gut, and physoclistus if the gases in the swim bladder arise from the circulatory system. The ventral lung is found in case of Sarcopterygi, and this ventral lung has been found to contain the internal divisions like that of into the alveoli or bronchioles. Now coming to the terrestrial organisms. Now the first terrestrial organism that was of that an amphibian, which was partly terrestrial and partly aquatic in its habit. In this organism, as I told you, there are external gills present at the larval stage in case of anurans, but in case of urodeles, that is the urodela, sometimes there are the external gills present in the adults as well. Internal gills are not present in the adults in this case, and internal gills are actually replaced by the skin respiration in their case. These amphibians have got the positive pressure pumps, that is, the energy is required for pumping the air into the lungs. What is the mechanism? Mechanism is as follows: they lower the pharynx, air enters into the pharyngeal region of the body via the nostrils, external nostrils, which communicate with the internal nostrils and then into the pharynx. From there it enters into the glottis, from there it goes into the lungs. In their case, lungs are very simple. Here the point to remember is that the internal nostrils came into existence for the first time. The exhalation follows the same course in their case. Amphibians have got 3-chambered heart, and in their case the double circulation came into existence for the first time, which means that for one circulation blood moves to the skin and the lungs for the oxygenation, and in the second circulation this oxygenated blood is taken to the entire body, entire systems of the body for giving the oxygen and taking the deoxygenated blood in return. One important point here of the comparative importance is that in case of fishes this was not the case, and they were having only single circuit, that is, there was only single circulation of blood. Circulatory system in these amphibians is again of very importance, because it is what actually enhances the rate of respiration, because these amphibians are comparatively at a higher metabolic rate than that of the fishes. What is the case with reptiles? There is no cutaneous respiration at all. That means skin is not vascularized, skin is covered with keratinized scales, etc. in their case. The ventilation system, that is breathing system or inhalation system, is the same. Trachea pulls the air into bronchi, from bronchi to the secondary bronchi, from secondary bronchi into the air sacs. These have got negative pressure pumps in comparison to that of the amphibians, which had positive pressure pumps. Here the energy is not required for inhalation purpose particularly. In case of birds, there are lungs, but these lungs are not elastic, but they have got the most efficient respiratory system, because they have developed air sacs in their body whose branches may run into the air spaces in the body, that is in the viscera, and also in the bones. These air sacs have got very important role to play in the respiratory system of birds. The number of air sacs in these birds is from 6 to 12 pairs. The mechanism of respiration is that in case of birds there is twice inspiration and twice exhalation, so that the air that enters once into the body leaves with maximum or 100% oxygen exchange with the body, and 100% carbon dioxide is thrown out of the body, which is not the case with the mammals or other vertebrates. The inspiration pattern that is followed by the birds is as follows. Inspiration 1, the air enters via the nostrils into the mesobronchus. From there, the air enters into the caudal air sacs, which is followed by the expiration 1 or exhalation 1. From the caudal air sacs, air enters into the mediodorsal bronchus. From there into the parabronchi. In inspiration 2, from the parabronchi, the air enters into medioventral bronchus. From there, the blood, this air enters into the cranial air sacs. From the cranial air sacs, the second expiration takes place via the trachea and the air is thrown out. This has been done to optimize and to make it possible the 100% exchange of oxygen with that of the blood. And at every stage of the bird, lungs and air sacs, the counter current mechanism of the flow of air and the blood has been there, which is for the purpose of optimizing exchange of gases. Now let us talk about the lungs. In case of lungs and the ducts that are associated with it, these structures are found only in case of terrestrial organisms. Lungs are elastic and distensible structures and have actually originated from the last pair of gills and gill pouches. Lungs receive the blood from the sixth aortic arch. The structures that are associated with the ducts of the lungs are: number 1 is the larynx, the trachea, the bronchi, the secondary bronchi, and ultimately the alveoli. In alveoli, the actual exchange of gases takes place. Rest structures are just of the conducting importance. Larynx came into existence for the first time in case of amphibians. They were simplest in case of Necturus, and the cartilaginous structure like arytenoid and cricoid cartilage came into existence in these organisms. Other amphibians have got simplest structure of this larynx, but in mammals it reached the maximum differentiation, where it is made up of 4 cartilages: thyroid cartilage, cricoid cartilage, and arytenoid cartilage. The arytenoid cartilage is present in a pair, that is, there are 4 cartilages in all. And in larynx also, in case of mammals, there are present vocal cords. These vocal cords help in creating the sound in case of mammals, and that is how that's why larynx is also referred to as the vocal box of the body. Instead of larynx, there is a structure called syrinx in case of birds, which help them in creating sound. Thus, in larynx, there are no vocal cords in case of birds, and it is soundless. But in birds, syrinx is the structure which creates the chirping sound in them, and they have got certain membranous structures present there which create the sound. Trachea is also called as the windpipe, which communicates the larynx with that of the lungs. This trachea is a structure associated with the glass cartilage, also called as hyaline cartilage. These cartilages are C-shaped structures which are not complete on the dorsal side of the body and are complete on the ventral side of the body, with certain exceptions among certain group of animals. These cartilages are there for a very important function to perform. What is that function? Is that the trachea is lined with the mucus, and in the absence of these cartilages, it would have been impossible to carry the air to the lungs, which would have otherwise collapsed in the absence of the cartilages. Larynx divides into 2 primary bronchi in the region of the lungs, and these primary bronchi enter the lungs, that is right and left lung respectively. In case of amphibians, the trachea is shortest, or sometimes it can be absent at all. Trachea is present in case of siren and amphibia, and there it is 4 to 5 centimeter long only. Reptiles have got varying lengths of the trachea, and its length depends upon the length of the neck. Longer the neck, longer will be the length of the trachea. In snakes and lizards, tracheal rings have been found to be complete. In case of birds, this trachea is elongated and is comparatively shorter than that of the reptiles because of the shorter length of neck, and it again has got the complete tracheal rings. In case of mammals, invariably these tracheal rings are incomplete. The exchange or the ventilation of the lungs is possible because of these ducts, and the passage of air will be from the internal nostrils to the larynx, from larynx to the trachea, from trachea air flows to the primary bronchi, from there to the secondary bronchi, from there to the tertiary bronchi, and ultimately they enter into the alveolar sacs, which are the actual sites of exchange of gases. Again here, the lining of the alveoli is the thinnest possible, that is the squamous epithelia, and the capillary network which is present there is again lined by the simplest squamous epithelia, which is referred to as endothelium, which optimizes the exchange of gases. Lungs are actually arisen from the swim bladder like structure, which was actually found in case of fishes, and it took a greater possible ventilation in case of mammals. Amphibians have got two lungs, simple lungs, which are hollow sac like structures. They are elongated and bulbous in case of Anura. Left lung is longer in case of Urodils. It is very rudimentary in case of limbless amphibians. In case of reptiles, particularly crocodiles, lungs are abdominal in position. Same is the case with that of the snakes, where the lung is tubular and is abdominal in position. Mammalian lungs are highly developed, spongy, elastic, and is divided into 3 to 4 structures, 3 to 4 lobes. The left lung is divided into 2 lobes only, while as it is 3 lobed in case of the right lung. In case of rabbit, the right lung is divided into 4 lobes. In the aquatic mammals like whales, the lung is very simple and without lobes. In the egg-laying mammals, right lung is highly lobulated. The bottom line of the study of respiratory system is that there are 4 important things to remember in the respiratory system. Number 1 is that the respiratory organ should have rich surface area, very high surface area, but for the increased surface area, the volume should be taken in relation. The second important point is that the respiratory surface or respiratory organ should be highly vascular. The third is that thinness of the respiratory surface should be very thin, very less, and very important point is the respiratory lining should be kept moist for optimizing the exchange of gases on the either side. Now friends, we have by now talked about the comparative structure of gills, the comparative structure of lungs, and the ducts that are associated with the respiratory system. We have also taken a brief idea about the evolution of respiratory system from the simplest possible to the highly complicated as is the case with the mammals. I will be concluding my topic here. Thank you very much.